Okay, so you're welcome to my video lecture on gene mapping. So what is gene mapping, ladies and gentlemen? A gene mapping is a process of determining the location of a genes on a chromosome. So we know that in humans there are over 25,000 genes, and each of these genes are responsible for producing a specific proteins. So you can actually know the location of a genes through a process of gene mapping on a chromosomes. We know that all these genes, they are actually located in the chromosomes of human. And of course, in humans, we have 46 chromosomes, although the number of chromosomes varies across different species. Like chimpanzee, they have 48. Drosophila melanogaster has 8. Donkey and other organisms, they have their own specific number of chromosomes. And in fact, is this chromosomes number and the different arrangement of the DNA sequence that make one organisms differ from one another. So the process of determining the location of a genes on a chromosomes is called gene mapping. So like, for example, in human, we have trillions of cells, and these trillions of cells, each of them have a nucleus, and this nucleus, like in humans, they contain a chromosome. And of course, the chromosomes in humans usually contain two chromatids. This is the first chromatid, and this is another chromatid. So generally in humans, we have a chromosomes, and in each of these chromosomes, there are two chromatids. So this is actually the two chromatids. And of course, in these two chromatids, we have what you call a centromere. And when we said a centromere is a center that actually joins the two chromatids together. So we can now decide to say, okay, we can take one chromatid and see. So if we take one chromatid like this is one of the chromatids. So the chromatid is divided into two. We have what you call a P arm and we have Q arm. So a P arm is the shorter arm, the chromosomes, while the Q arm is the longer arm of the chromosome. So you see here, the shorter arm is P, and then the longer arm is Q. So these are the two different arms of the chromosome. And of course, each of these arms is divided into region. Each of these arms is divided into region. Like, for example, let me demonstrate how these regions are. So you see, as I said, it has different regions. So let's have it like this. So we have a chromatid. This is a chromatid. And in this chromatid, we have what you call a regions like this. So this is called a region. And this region is also contains bands. It also contains bands like this. It also contains bands. It can be 2, 3, or 4. So this is 1, 2. So this can be 1. So you see we have 1, 2, 3 bands. And the band can also further be divided into sub-bands. They can also further be divided into sub-bands. So you see, this is the chromosomes. We have P arm here, we have Q arm, and this is the center where the chromatids are divided into two. And of course, we have the region, which also contain different band. So assuming now you have a address like this, you have, let's say, an address like this, 1 Q 2.4. So the 1 here stands for, it means chromosome 1, and the Q here means the long arm, and then the 2 here stand for region 2, while any number after point is the subbands. So it means that chromosome 1, Q-arm, region 2, and subband 4. So each and every chromosome, each and every genes in our body, it has a specific location. So ladies and gentlemen, let me just give you this as an assignment, and I want the answer to this on my comment section after you watch this video. Please, we have a gene called FOXP2, FOXP2. FOXP2 is a gene that allow us to talk. So we talk as human because we have this FOXP2. So now I wanted you to go and give me this answer. Where is the location of these genes? I want the address of these genes, the chromosomes number, the arm, whether it is long or short, the region, and the band or subband. Let me also give another example. Let's say we have an address like this 13Q, 14.3. So this is not 14, it's 14. So 13 stands for chromosome 13. Q stands for the long arm. The first one stands for region 1, and the 4 here stand for band 4, while the 3 here stand for subband 3. So this is how you give address to the gene. So ladies and gentlemen, in a simple term, when we say gene mapping is a process of determining the location of genes on a chromosomes. And this gene mapping, it helps in understanding genetic disease and traits by identifying specific genes with or associated with them. So each and every disease, like for example, let's say astigmatism, this gene mapping is actually very important, or it actually aids in identifying the functions as well as the location of genes that are associated with a particular character or with a particular disease. Like for example, astigmatism. Astigmatism is a disease that affects our eyes. So ladies and gentlemen, I wanted to also, this one is another assignment. Please, what is the names of the genes? There are 3 genes that are associated with the astigmatism. Please mention the genes and their address. That is their chromosome number, whether they are at the Q arm or T arm, their region, sub-region, and their bands. So this is what I wanted us to understand. So this gene mapping, it actually helps scientists to know the functions of so many genes, and also it helps scientists to identify genes that are associated with a particular disease. Like sickle cell anemia is a genetic disease, and there are genes of hemoglobin. So you can actually know the location, the region, as well as the band in which the genes of hemoglobin the genes of the hemoglobins that is found. So this is what you call gene mapping. So it's a process of identifying or determining the location of the genes, and it actually helps in understanding the diseases as well as the traits, as well as the characters or traits that are associated with a particular genes. So this field of mapping is an area that combines the genetics, molecular biology, and bioinformatics to analyze genetic data. So that is why, ladies and gentlemen, as a student of life sciences, we need to have a good understanding of genetics, molecular biology, bioinformatics, so that it will actually help in determining and analyzing the genomic data. So I think clearly this one is clear. That is, first here we define gene mapping, and then we also try to locate the address of the genes. So then the next thing that we are going to look at is what are the importance of gene mapping. So gene mapping is crucial for identification of genetic disorders. So there are many diseases that are associated with the genetics, like astigmatism, myopia, heart diseases, cystic fibrosis, cancer, sickle cell anemia, heart diseases, diabetes. All these are the genetic disorders. So gene mapping is very important in trying to identify the genes that are associated with these genetic disorders. And it also aids in the discovery of gene functions and their interactions. So there are so many genes that their function is not known, but it is this gene mapping that help in identifying the functions of the genes and also their interactions. So ladies and gentlemen, very importantly, as actually we discussed on some of my lectures, that genes are responsible for producing a particular proteins, and the proteins is what determine functions as well as the structure of some of our or most of our cells. So therefore, if you are able to identify the locations of the genes, you can definitely be able to trace the functions of the gene. So understanding gene location can enhance breeding programs in agriculture. So generally we have so many crops, we have so many agricultural products that some of them they are actually affected with pest, some of them they are affected with stress. So the knowledge of gene mapping, it can actually help scientists improve the breeding program so that you can have a products or agricultural products of high quality as well as the desired yield. So, and of course, gene mapping is very important. It's like a recombination between linked genes located on the chromosomes involves homologous crossing over. So, ladies and gentlemen, one very important things we can have chromosomes we can have, because when we say genes is an actually alternative form of genes. Usually they are in two form. So some of them they can be in the same chromosomes, some of them they can be in different chromosomes, or they can be at different arm. So the combination between the linked genes is actually one of the area that gene mapping delved into. So, of course, recombination changes the allelic arrangement on homologous chromosomes. So, generally, during genetic recombination, that is during meiosis, some of the genes can actually be located or can be actually translocate from one chromosome to another. But most of the genes that are very close to one another, they don't actually usually separate during the reproduction. They are actually inherited together from parent to their offspring. But some of the genes, they are far apart. Like, for example, let's say you can have a chromosomes like this. You can have a chromosomes like this. So you see you have chromatids. So you can have chromatid like this. So you can have genes here. You can have genes in this arm. You can have genes here. You can have genes here. So you see they are not closely together. And you can have also another genes here. You can also have another genes here. So of course, during genetics recombination, the genes in this chromatid can be actually transferred to this chromatid, and from this one, it can also transfer to here, and this one will go back to here. So it's possible during genetic recombination, but most of the genes that are very closely together, they usually not actually separated during reproduction. They usually actually transmitted to the young ones during cellular reproduction. So then the next thing that we are going to look at is the different types of gene mapping. So ladies and gentlemen, there are three main types of gene mapping. We have linkage mapping, we have physical mapping, and then the last one we have functional mapping. So the linkage mapping examines the inheritance patterns of genes in families. So we have generally we have genes families or family of genes. Some of the genes they don't exist as mono, they exist as a family. So the linkage mapping is a types of mapping that examine the inheritance patterns of genes in families. And of course, when we say physical mapping, is a types of mapping that focus in determining the actual distance between genes on a chromosome. And we also have cytogenetic mapping. So cytogenetic mapping is a types of mapping that usually use to stain the chromosomes and then visualize with a microscope. So, and it's usually when you use the staining or when you stain the chromosome, it will give different band when visualized with electron microscope. So, ladies and gentlemen, these are the two types of mapping here that we discussed. Linkage mapping is trying to determine or is trying to examine the inheritance of patterns of genes in families. So there are so many genes that actually came in family. You can have them, the families of the genes can be on the same chromatid, they can be at different chromatid, they can be at the same arm, they can be at different arm. So these types of a family of genes can actually be examined using the linkage mapping. But for the physical mapping, it's trying to focus on the actual distance between genes on a chromosome. So then this is actually a typical examples of linkage mapping. So in linkage mapping uses genetic markers. It uses genetic markers to determine the relative position of the genes. So generally, linkage mapping cannot actually precisely tell you the exact locations where the genes is. Rather, it gives a probability. It's a probability. It just gives a relative positions. And this linkage mapping, it relies on the principles of recombination, or it actually relies on the principle of recombination frequencies during meiosis. So the closer two genes are, the less likely they are to be separated during recombination. So if you have genes that are very closely together or that are very close to one another, so there are likelihoods that those genes, they cannot be separated during recombination. So it's only genes that are far apart or that are distant from one another that can be actually inherited. Therefore, the genes that are actually far apart, then there are likelihood that they can be separated during genetics recombination. So now, what are the genetics markers that are used in determining the relative position of the genes in linkage mapping? So these genetics markers, they are specific sequence in the genome that can be used for identification. And some of the examples of these gene markers include SNPs, that is single nucleotide polymorphism. We are going to talk about that later. And then we have microsatellites. So these are the examples of the genetic markers that can be used in tracing or in identification of the relative position of the genes in linkage mapping. So these markers, they actually facilitate the mapping processes by providing reference points on the chromosomes. So they are actually facilitating the mapping by providing the reference point on the chromosome. So that is why in linkage is just to actually determine the relative position. So then the next one is physical mapping. So physical mapping is a mapping that, as I explained, that is a mapping technique that can be used to determine the distance of two genes on a chromosome. And this physical mapping is conducted using techniques like FISH, that is fluorescent in situ hybridization. So fluorescent in situ hybridization is a techniques that use is a techniques get it is a techniques that uses fluorescent probes to identify and map a DNA sequence in a cell. So it's when you are using a fluorescent probes, fluorescent fluorescent probes to identify and map the DNA sequence in a cell. So this method helps pinpoint the exact location of the genes and their genomic features. So then, so the fluorescent probes used in this provide a high resolution view of the chromosome structure, which which easily help in identification of the genes. So this method helps pinpoint the exact location of the genes and their genomic features. So you see in this physical mapping, as I said earlier, the physical mapping is very important in the sense that it can actually pinpoint the exact locations of the genes and their genomic features, while the linkage mapping doesn't do that. So then the next thing that we are going to look at is functional mapping. So in functional mapping, it connects genes locations to biological functions, and the phenotypes. So in functional mapping, you can identify the location of the genes and their biological functions as well as their phenotypes. So in functional mapping, one, you identify the location of the genes. Then after identifying the location of the genes, then based on the locations, then the functions as well as the phenotypes of the genes can be traced. Like for example, if you can see clearly here, these are black animals, so it might be actually associated with their functions. So this is associated with the locations of their genes. So this approach, it integrate data first from genomics, that is the DNA, then that the total genome of a particular living organisms or a cell, and then transcriptomics, that is the RNA, and then proteomics. So it means that the functional genomics integrate the central dogma of molecular biology, because the genomics is the DNA. So then from the DNA, then the DNA will be converted into RNA, and then the RNA will be converted into protein. So you see, if you look at this, ladies and gentlemen, you can see that these are the three omics approach that can be actually integrated in functional genomics. So this approach is crucial for understanding the complex traits influenced by multiple genes. So in a simpler term, when we said functional mapping is a type of mapping that trying to connect the location as well as the biological functions and the phenotypes. You can have a black color, and it may be as a result of the your genes, which is melanin. So melanin gene is responsible for the black color. So the higher the concentration of the melanin, the higher the black color. The lesser the amount of the melanin, then the fairer the color. So this is actually what the functional genomics is. It's trying to, the functional mapping is trying to connect the location of the genes to their biological functions and their phenotype. So it is crucial in trying to understand the complex traits influenced by multiple genes. So then the next thing is, then what are the applications of this gene mapping in medicine? So gene mapping plays a vital role in personalized medicine by identifying genetic predispositions. So understanding the map of the genes can help or can assist in personalized medicine, especially in precision medicine, stratified medicine, and pharmacogenomics. So in precision medicine, is a type of medicine that use the genome, epigenomics, exposure, and other data to define individual pattern of disease, potentiality leading to better individual treatment. So you see, in precision medicine, you use the genomic data of an individual, just an individual, the genomic data, the epigenomic exposure, as well as the other data, especially like metabolomics, to understand the patterns of the individual disease, which potentially leads to a better disease treatment. So you can use the genomic data, my genomic data, like I as Mukhtar. You can use my genomic data, my epigenomics exposure, my metabolite to understand the disease pathogenesis. And then from there, by understanding my disease pathogenesis, then you will now develop a treatment that is specific based on my data that are generated. So this is actually precision medicine. It's a types of treatment that is applied to only one individual based on the genomic data. And then stratified medicine. In stratified medicine is the division of pattern of patients into disease subgroup based on predefined characteristics linked to the expected response to a particular therapy, their decreased risk of side effects from a particular treatment. And then, of course, in pharmacogenomics, which also part of the personalized medicine, is study or you study the genes and their functions to support the development of effective self-therapies that will be tailored to a person's genetic makeup. So actually in pharmacogenomics, you use genomic data as well as the functions of the genome to actually develop a particular treatment and therapies to a specific individual based on the data generated. So it aids in the development of targeted therapies for genetic disorder. So understanding gene interaction can lead to better prevention strategies for disease. So this is actually how you can apply the knowledge of gene mapping in personalized medicine, especially in precision medicine, stratified medicine, and pharmacogenomics. And then gene mapping in agriculture. So in agriculture, gene mapping is used to enhance crop traits like yield and disease resistance. Like, for example, one of the things that scientists have done is that they realize that there are so many insects that usually feed on our crops. So what scientists did actually, they tried to map a particular gene from a bacteria Bacillus thuringiensis. They isolate the gene and insert it into our crops. And then once the insect feed on that crops because of the inserted gene, will die. So that is how actually the use of gene mapping assists or helps in increasing crop yield. And also in addition, it allows for the identification of genes that are responsible for desirable characteristics in plant and animal. And it also assists in marker-assisted selection. So a marker-assisted selection is a technique that benefits from gene mapping data. So ladies and gentlemen, without taking much of our time, I'm going to actually stop here by understanding how gene mapping is important in medicine as well as gene mapping in agriculture. So ladies and gentlemen, if you have any questions, you can actually drop the questions on the comment section. And don't forget, if this is the first time you are coming to my YouTube channel, please subscribe and join my YouTube community for more videos. Thank you.